Okay, so what is the difference between cementoblast derived from dental follicle and those derived from Hedwig's epithelial root sheath? Um, what is it? So cementoblast derived from dental follicle. Okay, generally it will be like we'll say that's the old theory, isn't it? So cementoblast derived from dental follicle express the receptor express the receptor for PTH and extracellular proteins, osteopondin and osteocalcin. So they will express the receptor for PTH and extracellular proteins like osteocalcin and osteopondin. While those cementoblasts derived from Hedwig's epithelial root sheath do not express the receptor. Does not express the receptor for PTH and only express for osteopondin and partial osteoblastic expression. So those derived from dental follicle express the receptor for PTH and extracellular proteins like osteopondin and osteocalcin. Okay, we have this osteocalcin, and here you say osteoblastin's partial phenotypic. Okay, yeah, osteoblastic phenotype. Other one osteocalcin. Yeah. So what is the process by which cementoblast so what is the process? So what is the process by which cementoblast deposit collagen onto the dentine matrix? So cementoblast deposit collagen onto the dentine matrix by the process of mineralization, which actually allows intermingling of fibers of the two tissues at the future dentino-cemental junction. Dentino-cemental junction. What is alternative hypothesis of origin? So it is actually from the epithelial mesenchymal differentiation of Hedwig's epithelial root sheath. Differentiation or transformation like that. Yeah. So what is the role of non-collagenous matrix proteins in the formation of cementum? So non-collagenous proteins accumulate among in the spaces between fibers, and they help in mineralization. I don't know what, like mineralization. They have help or significant. They have a significant role. They have got non-collagenous proteins deposited into spaces. Non-collagenous proteins. Non-collagenous proteins deposited into spaces between fibers and play a significant role in mineralization. Okay, so what initiates the mineralization of cementum? Yeah, so this what initiates it? So what so we have mineralization of cementum. So what initiates it? So calcium binding amino acids. A calcium binding amino acids known as GLA protein. Then we have osteopondin, osteocalcin and osteonectin act as osteocalcin and osteonectin act as nucleotide substances to so osteocalcin and osteonectin. Okay, so what initiates the mineralization of cementum? So a calcium binding amino acids known as GLA protein. GLA protein. Then we have and osteonectin and osteocalcin act as nucleating substances and initiate mineralization. And this bone silo protein actually promote mineralization. Bone silo protein promote mineralization, and osteopondin regulate crystal growth. Okay. Yeah. So what does mineralize? How does mineralization advance in cementogenesis? Along collagen fibers. So mineralization advance. Along collagen fibers without matrix vesicles in cementogenesis. So mineralization advance along collagen fibrils without matrix vesicles in cementogenesis. Okay, in cementogenesis. So what triggers the mineralization process? What triggers the mineralization process during initial cementogenesis? So calciferous spirules formed by matrix vesicles triggers the mineralization process during initial cementogenesis. So what triggers mineralization during calciferous spirules? So calciferous spirules produced by matrix vesicles released by cementoblast actually trigger mineralization during initial cementogenesis. So what triggers mineralization during initial cementogenesis? This calciferous spirules formed by or released by matrix vesicles released by cementoblast triggers mineralization during cementogenesis. That is during initial cementogenesis. So calciferous spirules formed by matrix vesicles released by cementoblast initial mineralization during triggers mineralization during initial cementogenesis. So what happens to cementoblast as cementum formation proceeds? What happens to this so-called cementoblast? Maybe get embedded? No, not embedded. That's like no. They recede. So what happens to the cementoblast as the cementum formation proceeds? They recede outward, lining the periphery of the outer surface of the cementum. So what happens to the cementoblast during what happens to cementoblast as cementum formation proceeds? The cementoblast recede outward and line the periphery of outer surface of cementum. Line the periphery of outer surface of cementum. Line the periphery of what? Line the periphery of outer surface. Line the periphery of outer surface of cementum. What is the difference between? So based on the difference between there are two types of. So what is the difference between two types of cementum in terms of receptors expressed on cell surface? So based on so there are two types of cementoblast in terms of receptors expressed on cell surface and their reaction. So there are two types of cementoblast in terms of receptors expressed on cell surface. Okay, there are two types of cementoblasts in terms of receptors expressed on cell surface and their reaction to signaling. So there are two types of cementoblast in respect to receptors in respect to receptors expressed on with respect to receptors expressed with respect to receptors in terms of receptors expressed on cell surface and their response to and their reaction to signaling. In terms of receptors expressed on cell surface and their reaction to signaling. So in terms of what? In terms of receptors expressed on cell surface and their reaction to signaling. There are two types of cementoblasts. Suggest about the epithelial mesenchymal transformation in Hedwig's epithelial root sheath. Yamamoto. So investigations by Yamamoto et al. suggest the epithelial mesenchymal transformation in Hedwig's epithelial root sheath. So investigations by Yamamoto. So investigations by Yamamoto et al. Suggest about the epithelial mesenchymal transformation in Hedwig's epithelial root sheath. So what is the origin of cementoblast according to original hypothesis? That is the mesenchymal cells of dental follicle. According to original hypothesis, dental follicle is the origin of cementoblast. What is the origin of cementoblast according to original hypothesis? According to original hypothesis, dental follicle is the origin of. According to original hypothesis, dental follicle is the origin of cementoblast. What is the difference between cementoblast? So cementoblast derived from dental follicle respond to PTH and other proteins, proteins like osteocalcin and and while those derived from the Hedwig's epithelial sheath does not respond to PTH and respond to and only respond are expressed for osteocalcin, and then you have partial osteoblastic phenotype. Under the osteocalcin, I think osteocalcin for dental follicle is osteocalcin and osteopondin. Yeah, then you have osteocalcin and osteoblastin. Osteocalcin and osteopondin. Yeah, it is osteopondin, osteocalcin, osteopondin, osteoblastic. Osteopondin is the common one. Okay, osteopondin, osteocalcin, osteopondin, osteoblastic. And it is a extracellular. These are extracellular. What proteins? These are extracellular. Express the receptors for PTH and extracellular proteins and extracellular proteins, that is osteopondin and extracellular proteins, osteopondin and osteocalcin. What is the conclusion of Yamamoto and Takahashi study on Hedwig's epithelial root? What was the conclusion of Yamamoto and Takahashi study on Hedwig's epithelial root sheath cells? Yamamoto and Takahashi studies concluded that Hedwig's epithelial root sheath cells do not transform into cementoblast in rat molar cementogenesis. Hedwig's epithelial root sheath cells does not transform into cementoblast in rat molar cementogenesis. That was the conclusion of the studies of Yamamoto et al. And Takahashi studies on so that is Hedwig's epithelial root her cells does not transform to cementoblast on rat molar cementogenesis. What is the role of non-collagenous matrix proteins in the What is the role of non-collagenous matrix proteins formation of cement? So matrix proteins are deposited in spaces between the fibers and have a significant role in mineralization. What is the difference between acellular and cellular cementum? So cellular cementum is actually formed at a faster, the rate of formation of cementum is at a faster rate, and you have few cementoblasts embedded. And acellular will be very slow rate and more cellular will be more slow rate. What does mineralization how does mineralization advance during cementogenesis? During cementogenesis, how does it advance mineralization? So mineralization advance along collagen fibrils without matrix vesicles during cementogenesis. That is how mineralization advance during cementogenesis along collagen fibrils without matrix vesicles. How does mineralization advance during cementogenesis? Along collagen fibrils without matrix vesicles. What initiates mineralization of cementum? So we have calciferous spicules formed by matrix vesicles released by cementoblasts. Calciferous spicules maybe. I think spicules, calciferous spicules released by formed by matrix vesicle released by cementoblast. What initiates mineralization of cementum means it is a completely different answer. Okay, like maybe it's like how does what triggers during initial cementogenesis. This is what initiates mineralization cementum. It is the calciferous calcium binding proteins like calcium binding amino acid GLA protein and osteoblastin, no osteoblastin. Okay, we have osteopondin. Some other answer we have osteopondin, osteo calcin, osteopondin, osteoblastin. Here you have osteonectin and osteocalcin. No osteobondin. Okay. Here we have osteocalcin, and we have this extracellular proteins like osteocalcin, osteocalcin, and osteonectin. Yeah, they act as what? They act as nucleating substances. They act as nucleating substance.